Hello, the topic which we are taking today is genetic engineering. So it's from genetic engineering including in genomics. So we have genetic engineering. So let's write the topic. That's in fact genetic engineering. So genetic engineering is a biotechnology involved in synthesis of artificial genes, repair of genes through fusion, deletion, inversion, shifting, which is also called transposition. So shifting of genes, production of recombinant DNA, that is combining DNA from two sources and manipulating them for improvement in human beings. So artificial gene, we have repair gene, then which is called a recombinant DNA technology. So that's actually and manipulating them for improvement in human beings, plants, animals, and microbes. An important aspect of genetic engineering is recombinant DNA technology, in which desired DNA sequences from different sources are obtained and joined together to form a new combination or recombinant DNA or rDNA. So all the three is important. This rDNA gives a desired product or trait. This is important. Now, recombinant DNA technology is also called a gene splicing, as it involves cutting and pasting of desired DNA fragments. So basically, if we take all the aspects of genetic engineering are very important, and rDNA technology is most important because of mixing of different genes. So this gene splicing, as we call it, it's a simply cutting and pasting of desired DNA fragments. So it is based on two important discoveries in bacteria. So we have product and we have trait of desired thing. Now, as I said, this giving product and getting trait, two important aspects of gene splicing or rDNA technology. It is we can say that if we take this rDNA technology, as I said, is a gene splicing method. So it is based on two important discoveries in bacteria. So we had two discoveries in bacteria which give rise to this all the phenomena, latest phenomena. The two discoveries where we can say those were the first was plasmid, this presence of plasmids, as discovered by Hayes and Liederberg in 1952 in bacteria, which can undergo replication along with and independent of chromosomal DNA. And second was restriction endonucleases, given by Arber, Nathan and Smith in 1970, got Nobel Prize in 1978. So restriction endonucleases, which can break DNA at specific sites. So they are appropriately called chemical scalpels or molecular scissors. So the science of recombinant DNA technology took birth very much before, when there was a scientist actually, Berg, in 1972, who was able to introduce a gene of SV40 into a bacterium with the help of lambda phage. So Berg is often considered, that's why he's considered as, father of genetic engineering. So he would introduce SV40 into bacterium with the help of lambda phage. That's why he is father of genetic engineering. And for this, he was awarded Nobel Prize in 1980. So he also got Nobel Prize 1980. Then we have Cohen and Boyer, where in 1973, two people who were able to introduce piece of gene containing foreign DNA into plasmid of Escherichia coli. So were two important scientists who were able to do this. So now recombinant DNA technology involves 5 steps actually. The first is that is separating a desired useful treatment segment from an organism called a donor organism, selection of suitable vector, that is vehicle DNA, and fusing the selected DNA segment, that is passenger DNA, to it to create a recombinant DNA. Now these are 2 steps: getting the desired DNA, getting vehicle or vector, and then fusing them together, fusing that segment of DNA into this vector, which is in fact a plasmid. Then third is this producing numerous copies of recombinant DNA, that is cloning of this mixed DNA or mixed, that is desired DNA and vector, making copy of it. This is the third important step. Now we are going to take the fourth step, which is very important, that is inserting the recombinant DNA into cells of recipient organism. So after making a lot of copies of these recombinant DNA, we are going to move into the another segment, that is inserting the recombinant DNA into cells of recipient organism. What that can be depends upon the choice or the method, the procedure, or the economy, the steps where you want to have them. So this is introducing this into this recipient organism. Now fifth can be followed, there is screening. So after this we have to move on to the fifth step, and that fifth step is screening the transformed cells for desired change and finding economical method for mass production of gene product. So after completing these 4 steps, we're gonna to move to the 5th step, and that's very important. As I said, the science of genetic engineering and recombinant DNA technology is being used for different things. So we have only these 5 steps. This 5th one is screening the transformed cells for desired changes and finding economical method for mass production of gene product. Now this science of genetic engineering and recombinant DNA technology, it is being used for different things. If we take it is usage of genetic. First is understand molecular events in biological processes like growth, differentiation, aging, etc. Second is preparation of gene maps. And the third is reversing or checking the process of development by identifying the genes and modifying them. The fourth one is finding out the complete nucleotide sequence of a genome of an organism and identification of genes. And that's very important because we gonna identify genes of different organisms. That is the fourth one. And as I said, 1, 2, 3, 4 methods. The fifth one is study of defective genes. That is the fifth one. Study of defective genes. Then we have diagnosis of many diseases like hepatitis, HIV, cystic fibrosis, food poisoning by salmonella and staphylococcus. So here a short segment of single-stranded DNA attached to the fluorescent or radioactive marker is used. Okay. Now we are going to take another important aspect. It is used for identification of genetic disorders and finding out remedial measures through genetic counseling. And the eighth one is production of genetically modified organisms or GMOs or transgenic organisms providing particular product and nutrient. Then synthesis of important biochemicals and pharmaceutically important products with the help of genetically altered genes. Okay, so many functions, so many uses of this recombinant DNA. We have, with the help of, we have found human insulin or Himalayan treatment of insulin-dependent diabetes, human growth hormone treatment of short stature, calcitonin treatment of rickets, chorionic gonadotropin, blood clotting factor, tissue plasminogen, erythropoietin, platelet-derived growth factor, interferon, interleukin, vaccine, removal of defective genes. So many uses of DNA, genetic engineering. So now we have one more remedy. There's gene therapy. So it's a removal of defective genes and the replacement with the normal, healthy, in functional genes. That's called gene therapy. For example, sickle cell anemia, severe combined immunodeficiencies. There is SIDS. In SID, 25% of cases there is a defect in gene for enzyme adenosine deaminase, that is ADA, and functional T lymphocytes are absent or immune response to the pathogen is absent. Stem cells from the bone marrow are extracted from the patient. Copy of the human gene encoding for the enzyme ADA is introduced into the lymphocyte with the help of viral vectors. The cells come to have both the defective and functional genes. The treated cells are injected to patient's bone marrow. As they become operational, they begin to form T lymphocytes and the patient's immune system becomes natural. Now, SID correcting is one of the important beginning discoveries of genetic engineering. That was one of the important discoveries, one of the important therapies, one of the important gene therapy in fact. Now, tools of genetic engineering. What are the tools? How we use the method of genetic engineering? The first is source of desired gene. Now it can be source of desired gene, culture of cells, synthesis of artificial gene with the help of gene machine, purified mRNA, and reverse transcription, gene amplification. The desired DNA to be passed into cells is called passenger DNA. So the tools for sources. Now second is DNA modifying enzymes. So we have restriction and denucleases, DNA ligases, DNA polymerases. So many of enzymes are used. We have third cloning vectors. They are carriers of passengers or desired genes. Vectors also possess their own DNA, which is called vehicle DNA. Vehicle DNA carrying passenger DNA is called recombinant or rDNA. There is chimeric DNA. Now cloning vectors include plasmid, plasmid, bacteriophages, this viruses parasitizing bacteria, plant and animal viruses, transposons, jumping genes, and art. So, as I said, that's how this vectorless or directed transfer of desired gene can also be carried out through electroporation or chemical induced transformation, microinjection, and particle gun methods. So many methods are there for this. That's cloning vectors. Now we have artificial synthesis of gene also. So artificial synthesis of gene, it was actually scientist Khurana et al. This 1968. They prepared the first artificial gene of alanine tRNA having 77 nucleotide pairs. 77 nucleotide pairs. Basically, however, it did not function. So, the first functional artificial gene was tyrosine tRNA gene with 2207 nucleotide pairs, also by Khurana et al. 1979. So it was a functional one. In 1981, Edge et al. Synthesized IFN alpha gene, that is human leukocyte interferon gene. He was the important discoverer of this. So, but the base pairs were with 514 base pairs, interferon base pairs. So we can say that's IFN alpha gene, lymphocyte interferon gene, 514 base pairs. Then insulin A, insulin B, and somatostatin genes have also been prepared. Now there is a one more important, that is gene machine. Now if we take gene machine is now available with which help us in linking nucleotide and specific sequences. So this is a gene machine, help us nucleotide to link in a specific sequence to give a gene of, then followed by PCR, etc. whatever we see. We have PCR tag polymerase, and so then much more than this. So we have reverse transcription, we have gene amplification through polymerase chain reaction, 72-degree temperature to 90-degree, tag polymerase, Thermus aquaticus. This is obtained from hot bacterium. With this we complete this genetic engineering, a little topic. Thank you for watching.